this is an overview. The beginning of understanding how the kidneys work is simply how they control their own filtration, their own specific intrinsic function, which is the filtration rate. And this happens through three main methods. These three methods, if you're the astute student, will notice that these three methods of regulation are recurring throughout the semester. For example, we could say the same thing about the heart, or we could say the same thing about the GI tract. These are the three sort of general ways that an organ can be regulated. There's going to be this auto-regulation, the regulation at the local level using either autocrine or paracrine hormones, specifically in this case paracrine hormones. And do you guys remember what paracrine hormones mean? Do you remember what that term means? I ran it by you in the beginning of the semester when we were talking about glandular epithelia. So paracrine is a hormone that is produced by one cell and then released for the benefit of its nearest neighbor. All right, the cell right next door. It doesn't necessarily go through widely distributed throughout the circulatory system. So there's this auto or local level to the auto regulation, to the control of the glomerular filtration rate. Then there are the chemical and the electrical, the hormonal regulation that is indeed initiated by the kidneys, and there's going to be significant innervation by the sympathetic autonomic nervous system. I don't know, I sometimes regret a little bit that I didn't go through the autonomic nervous system to any appreciable degree this year. I did last year, but it's enabled me to cover what I cover a little bit better, I think. So we'll touch on the autonomics lightly and mostly focus on the hormonal and a little bit of the auto-regulation of the GFR. The regulation of the GFR is related to other functions of the kidney, specifically blood pressure. Okay, we'll get to that in a bit. So here's a homeostatic cycle for the glomerular filtration rate. I'm going to show you two of them. Normally when you see these homeostatic cycles I put up on the board here, they're like a figure eight, right, where the homeostasis is in the middle and there's some variable that's being monitored, and if it rises, the loop goes one way, and if it falls, the loop goes the other way, right? So you can imagine this as being one half of that figure eight, all right? If the glomerular filtration rate, if we're going to disturb normal GFR and we increase it, this is going to lead to an increase in the production of urine. All right, so if there's more filtrate, there's going to be more urine, and if there is more urine, it is going urine production. That's going to be sensed by the macula densa. Do you guys remember what the macula densa was? Yeah, it was part of the juxtaglomerular complex. So let's remember the nephron for a moment. We have the renal corpuscle where the glomerulus is and the filtration happens, and it goes into this tubing, the proximal convoluted tubule, down the loop of Henle into the distal convoluted tubule, and that distal convoluted tubule loops back and intersects with the vascular pole, where the afferent and efferent capillaries are. This region right here is the juxtaglomerular complex. This is sort of a sensing and integration center. All right. So if there's increased urine production, the macula densa is going to recognize that, and it's going to interact with the juxtaglomerular cells, which are going to initiate paracrine secretion. All right, there's going to be hormone production. That's going to result in a number of effects. So first of all, the afferent arteriole is going to constrict. This means it's going to limit the amount of blood that's getting into the glomerulus. Right? You can see how that would reduce the glomerular blood pressure and then consequently the amount of filtrate that's produced. Likewise, the mesangial cells are going to contract, and you'll remember again that the mesangial cells are these contractile cells that kind of wedge themselves between the capillaries of the glomerulus itself. And when they contract, that's also going to restrict blood flow into the glomerulus and have a similar reduction in glomerular blood pressure and filtrate production. And then lastly is the dilation of efferent arterioles. So releasing the pressure head on the front of the blood so that less pressure is able to build up in the glomerulus itself. All right? And all of this will reduce the blood pressure in the glomerulus and then consequently reduce the filtrate production. Bringing us back and closing the homeostatic cycle. So in general, just a few statistics on the bottom. Your glomerular filtration rate is about 125 milliliters per minute. That's about a little juice cup of fluid a minute. That's quite a bit. It's quite a bit if obviously we're going to be resorbing most of that fluid. If your kidneys were pumping out that much urine every minute, we would be going to the bathroom all the time and be rapidly dehydrated. This represents 180 liters a day. 180 liters a day. That's a huge volume. It's a huge volume of fluid. The vast majority of it, as I just said, is going to be resorbed by the renal tubules. So about 10% of the fluid that enters the kidneys via the blood leaves the bloodstream into the capsular space. All right, now here's the other half of that cycle. So what if the glomerular filtration rate drops and we need to increase it? We're going to dilate the afferent arterioles, going to let more blood into the glomerulus, and relax the mesangial cells likewise, and then increase the pressure in the glomerulus by constricting how much of the blood can actually leave via the efferent arterioles. All of that will serve to increase the blood pressure, and since filtrate production is, yes? We'll talk about all of this in a minute. You just be patient. Yes, there is. Trust me. You just hang on to your hat. Okay. So, excellent question. This would seem to be pretty self-explanatory at this point. The blood pressure goes up in the glomerulus, more filtration is going to happen, and more filtrate will be produced, thus more urine. Again, all of this is going to be sensed by the macula densa. Anything else on this one? No. All right, so that is the control of glomerular filtration rate. There are a number of, so when I was going through the functions of the kidney, I talked not just about the control of fluid volume in the body, I also talked about pH and electrolyte balance, didn't I? So there is this sodium, potassium, and water homeostatic cycle that happens here at the kidneys. And there's a lot, it's pretty sophisticated. And I'm going to go through some of the aspects of that. And these, I'm going to introduce the players to the story. So these are the molecular hormones, the molecular factors. So they're going to be hormones and enzymes. The major hormones, the ones that I'm going to talk about quite a bit, are angiotensin. Angiotensin has three forms. The most important is angiotensin 2, aldosterone, and ADH. I'm not going to say what they do right now because that's the rest of the lecture. We'll get to it. There's a range of other hormones that are also players in the story. It would take probably a whole week for me to go, an additional week on the kidney to go through all that stuff, maybe more. And then there are these enzymes, renin, which is extremely important, and angiotensin converting enzyme, or ACE, which is also important in the story that I'm going to tell. So I'm not really going to talk about any of these ones in the middle, but they're also very important. Okay. Well, I call them minor, but they do play an important role. Okay. So here are some of the systemic factors that are involved in controlling sodium, potassium, and water levels in the body. So first of all is the GI system. This is how we're going to absorb all three of these things. We bring them into the body, and then some of them are secreted out into the GI tract. But mostly, this is about influx into the body. This fluid is that's in the body then is in exchange with the we're going to the other side here, the intracellular space. And we've talked about sodium and potassium, how important they are. Fluxes in sodium and potassium across the membrane are what give cells the ability to form graded potentials, and excitable cells the ability to have action potentials. There is an osmolarity that needs to be maintained within the intracellular space. So there's a significant amount of fluid and electrolytes that's actually contained and exchanged from the intracellular to the extracellular space across the membrane. Now the fluid and the electrolytes are going to leave the body primarily via the renal system, the kidneys here. We're going to filtrate a huge amount of these out and then resorb the vast majority of it as well. All of this is under the control of the endocrine system and the nervous system as well. I didn't put this one in there. I probably should put that next to the endocrine system, the control factors that are on top. So the hypothalamus, the pituitary gland, which are the central nervous system structures on the left up there, and then the adrenal cortex, which is what is depicted on the right of that. So these are the systemic factors that are involved in sodium, potassium, water homeostasis. Now, you can't really talk about sodium, potassium, and water regulation without talking about blood pressure regulation because they are intimately intertwined. In some ways, they're almost synonymous. So maintaining the appropriate levels of sodium, potassium, and water in the body is one of the largest ways that we're able to modulate blood pressure. There are others that are outside of this that I'm not going to touch on, but I'm going to focus on the relationship between sodium, potassium, water, and blood pressure, specifically the central role that the kidneys play in maintaining all of this. So then let's look at an overview of blood pressure regulation. Again, these are all just overviews. So here's this same motif that I've talked about. There's the auto-regulation. This is maybe the only slide that I've shown this semester that is a holdover from Andrea Tilden's class. She gave me all her materials, and this is the only slide of hers that I've used. But there is the auto regulation that I have talked about that uses various tissue-specific forms of local control. Then there is the nervous system, the hypothalamus, and the medulla are both involved in that. The medulla, we'll leave it there actually. The hypothalamus and the medulla are part of the central nervous system. And then in the periphery, there's a number of players as well. There are Barrow receptors. What's a Barrow receptor? Pressure receptor, that's it. There are Barrow receptors in the aortic arch and in the carotid bulb, all right, that are going to sense your blood pressure. And then there are sympathetic and parasympathetic ganglia that are going to innervate various effectors, specifically in the kidney, that are related to blood pressure. And then lastly, there's hormonal control. So there's the three big, and I showed you all those molecular factors. The three big categories are this Renin-Angiotensin-Aldosterone, what's called Axis. We'll see what that means in a moment. Still just setting everything up here. Then there's Antidiuretic Hormone. It also goes by the name vasopressin. And an important one is Atrial Natriuretic Hormone that is produced by the heart. And I'm not going to take, I don't think, maybe I will. I doubt I'm going to have a chance to talk about it at all. But it is an important one. ANH was on that list that I showed you a few moments ago. It's one of the many characters in the story. All right. So I need to also introduce this concept of an axis. An axis is a set of influences, various molecular factors, feedback mechanisms. It's a sequence of them that are going to connect various organs in a number of systems, primarily the endocrine system, and the target organs for those molecular factors. So the two that are the most important here for the blood pressure homeostasis are called the Renin-Angiotensin-Aldosterone Axis, RAA axis, and that's going to be what I spend probably the most of my time talking about today after the setup here. And then there's the hypothalamic pituitary adrenal axis, the HPA axis, and we will touch on that as well. So these two sort of corridors of control for blood pressure. And I don't want to pretend that these axes are fully independent of one another. They're not. They interact, and we'll see how they interact. So, pen still scribbling. Everybody, are there any questions about what I've gone through so far? If you don't understand, ask in class. In class is a good place to learn. Okay. So let's go over the RAA axis. What I'm about to put on the board can be thought of, again, as a homeostatic cycle. This is a cycle. And in some ways, it is an expansion. I'm just filling in the details on the same cycle that I showed earlier, all right, as it relates to the glomerular filtration rate. As we step through this, there's obviously in all this blank space, there's going to be some stuff that appears in a moment. I didn't want to overwhelm you with it all at once. The color key is in the green boxes. These are going to be some sort of physiological factor, all right. In yellow, they're the hormones or prohormones, angiotensinogen. This first box here is actually a prohormone. A prohormone is the same as like a proenzyme or a zymogen, if those words mean anything to you, except it's it's a hormone, not an enzyme. And then in blue are enzymes. These are the two that I showed you in the beginning when I pointed out the molecular factors. So let's begin. The liver constitutively produces angiotensinogen. In general, when you see the name of a protein and the name of the protein ends in something like ogen, that is a key, that's a tip to you that, that the protein in question is not in its fully active state. It's an inactive protein, and it's going to become, or maybe it has some kind of functionality, but there is an occult functionality, a hidden functionality that is yet to be revealed by some sort of regulatory process. Okay, when you talk about enzymes, they're called Zymogens. Trypsin is a protease that cleaves up other proteins, and trypsinogen is the inactive form, the precursor to it that's going to get activated by something. In fact, it's it activates itself. All right, so angiotensinogen is produced by the liver, and it's a pretty long amino acid, about, well, I guess a peptide. It's 453 amino acids long, comparatively long to the functional bit. The kidney, in response to a drop in blood pressure at the glomerulus, so there's a reduction, this is answering your question, there's a reduction in blood pressure at the glomerulus. Why? Maybe there's a reduction in blood volume. Who knows? Maybe you're menstruating, or you injured yourself, or who knows why. But there's a drop in blood volume for some reason. Maybe there is a drop in systemic blood pressure for other reasons. We're not sure. There's a host of reasons that could be. Perhaps pathologically there could be a renal artery blockage. That's serious. You don't want that to happen. The kidney will die, but it can happen, and you'll get a drop in blood pressure at the glomerulus. When this happens, the macula densa, which is part of the juxtaglomerular complex, it's the sensory part of the juxtaglomerular complex. It's the patch. A macula just means a round patch. You'll remember that term from the eye. I talked about the macula, which is the patch at the center of your field of view that the fovea is embedded in. The drop in osmotic pressure or concentration in the tubular fluid is picked up by the macula densa, and it causes the juxtaglomerular cells to produce renin. Renin gets released into the bloodstream. Renin is an enzyme that cleaves angiotensinogen. It has protease activity, or peptidase activity, I guess, and specifically cleaves a big chunk off of angiotensinogen, or the regulatory chunk that surrounds angiotensin and keeps it from doing its job, and releases this 10 amino acid chunk of angiotensinogen. Angiotensin, this is what we call angiotensin 1, 10 amino acids long. This continues to circulate through the bloodstream, and the lungs constitutively produce another enzyme called angiotensin-converting enzyme. Again, has a peptidase activity that is going to cleave 2 amino acids off of angiotensin 1 and give us angiotensin 2. All right, so this is where the fun starts. We've got angiotensin 2 finally. So let's take a first look at the rest of the Raa axis. All right, we have renin, we have angiotensin. How do we get aldosterone? Well, it turns out that angiotensin 2 has a catalytic effect on various steroid precursors that are produced in the suprarenal cortex. Suprarenal cortex, also known as formerly known as the adrenal cortex. These steroid precursors are converted by angiotensin II into aldosterone. And aldosterone is going to have a direct effect on various parts of the kidney. Now, this little cycle here, I'm sort of quaintly depicting it with a simple little green arrow, but there's a lot going on here. We're going to unpack that, exactly how that works. But the consequence is that there's going to be sodium and water retention. When we retain water in the bloodstream, when we pull more water out of the filtrate, that's going to raise our blood volume. And if we're going to raise our blood volume, we're going to raise our blood pressure. Okay? And if we also pull sodium back into the blood, that's going to cause an osmotic gradient that's going to help drive water retention. Okay? Water is going to want to follow the salt. And homeostasis will be restored. And increased glomerular, man, I'm having a hard time with that word today, filtration rate, GFR. You guys good with this so far? All right, let's add angiotensin II is a dynamo. It does a lot of things. Here's some more of what it does. It is able to have a direct effect on the circulatory system. So we're going to move from left to right across these other things. So it's going to affect vasoconstriction in the arteries in the vascular system, thus reducing the actual space that the blood is confined to, and consequently raising the blood pressure. Not directly raising blood volume, but this is an effect on blood pressure. Then there is the effect of angiotensin II on various neural structures. The first is on the hypothalamus, and this causes an increase in your sense of thirst and stimulates you to begin drinking. You're thirsty. And then angiotensin II also has an effect on the pituitary gland. The pituitary gland is a pretty sophisticated organ. We'll talk about it a little bit more in the endocrine chapter, but you can kind of think of it as like the peak of the pyramid of the endocrine system. All right, it produces a lot of hormones that control a lot of different systems and a lot of different organs. Well, in response to angiotensin II, the pituitary is going to produce antidiuretic hormone. This is also going to, I should probably have the arrow instead of pointing to the green box, I should have it pointing to the kidney. Because it does the same thing, though. It leads to sodium and water retention. And we'll see how in a bit. All of that, again, leads to elevated blood pressure and blood volume. So it doesn't look like a cycle, but this box down here would simply lead to homeostasis restored, and then if it was perturbed again, we go back to the green box. So that's the Raa axis. Oh, I forgot to point the last thing out. Angiotensin II also increases sympathetic activity. Sympathetic activity sends out sympathetic nerve fibers, stimulate contractile cells on the afferent arteriole directly. I couldn't fit that all in one diagram. So here is a little bit of a look now at, let's see, let me go back. We're gonna unpack this, the ADH and aldosterone effect on the kidney. All right, we're going to look at this a little bit more closely and see how it actually affects sodium and water retention. All right, so on the left here, let's just go over the general flow first. So we have low renal perfusion pressure, and we're going to produce the macula densa will sense that, will secrete the juxtaglomerular cells in response will secrete renin. This gets put into the bloodstream and leads to the production of angiotensin-2 and aldosterone via the pathways I just showed you. What are the effects? The effects are an increase in sodium resorption and an increase in hydrogen ion retention. So an increase in an increase in hydrogen ion export, pardon me, I had that backwards, and thus an alkalinization of the blood, an increase in the serum pH. The sodium reabsorption helps to drive water reabsorption by an osmotic effect, both active and passive effects, and this leads to an increase in blood pressure. So let's just walk along the nephron and see how this happens. We have the juxtaglomerular cells that, under the response of under the direction of the macula densa, will produce renin. That leads to through all the pathways I showed you, to the angiotensin-2 and the aldosterone. This should be aldosterone 2. I need to fix that. The first part of the nephron, after the glomerulus, is the proximal convoluted tubule. Angiotensin-2 leads to the resorption of sodium, and to keep charge balance, it also absorbs carbonate at the proximal convoluted tubule. So there's no net charge difference. It's simply in a change in osmolarity of the tubular fluid and a resorption of sodium. We're going to see how this works in a few slides. Proximal convoluted tubule is a little bit, it's got a lot going on, and we'll see how that works in a bit. Then there's the loop of Henle and countercurrent multiplication. We'll talk about that. And we get here to the distal convoluted tubule. Now, at the distal convoluted tubule, aldosterone, oh no, it's aldosterone, not angiotensin. My eyes are failing me here. So aldosterone is the main hormone in the distal convoluted tubule, and it's going to achieve two things. The first is on this side, the sodium resorption and water retention, and the other is in terms of hydrogen ion export. So the top bit here is the effect of aldosterone on what are called principal cells. There are these cells that exchange sodium for potassium. So it brings sodium out and a potassium into the tubular fluid. All right. So again, a net charge neutrality. And then these other cells called intercalated cells, the principal cells. So the names kind of tell you here, if you were to take a histological slice of this tissue, most of the cells in the distal convoluted tubule they're principally principal cells, and then intercalated between them are these few intercalated cells. The job of the intercalated cells is to take that potassium that was exported by the principal cells and use that in an antiporter. Both of these cells, principal cells and intercalated cells, are antiporters. It's going to export hydrogen ion and achieve charge neutrality, charge balance, by importing the potassium that, that was exported by the principal cells. Okay, you guys follow that? So angiotensin II works in the principal convoluted tubules to pull sodium and carbonate out, alkalizing the blood, acidifying the urine. And then on the distal convoluted tubule, it's aldosterone that works on the principal cells to resorb sodium and excrete potassium, and then some of that is resorbed by the intercalated cells as it exchanges it for hydrogen ion. Okay, so let's look in even more detail here at the principal convoluted tubule. Whoa, look at that diagram. There's a lot going on there. Luckily, I'm not actually going to ask you to memorize all the details on this diagram. I've already given you quite a bit. But there are some points that I want to make showing you this diagram. So there's multiple pathways in the proximal convoluted tubule. There are proteins that are going to absorb glucose and other organic solutes. Remember, this is the baby in the bathwater story, right? So all of the glucose and other organic nutrients get filtered out at the glomerulus. We don't want to lose those in the urine, so we want to pull them back in. That happens in with a co-transporter for sodium. And there's another sodium-involved transporter. It's actually an antiporter that exports hydrogen ion into the tubular fluid and again achieves charge parity by bringing a sodium across into the cell. All of this sodium, and there's just plain old sodium transporters as well. All of this sodium is used to is exported from the cell, and we import 2 potassium. So you've seen this enzyme before. You've seen this machine before, haven't you? You've seen this in neurons. Yeah, this is the sodium-potassium ATPase. This burns ATP is going to pump 3 sodium out and bring 2 potassium into the cell. There's another little bit of a sophisticated pathway here that's going to take bicarbonate and import it into the cell and eventually export it to the blood out of the tubular fluid. The point that I want to make here is that there is both the resorption of organic nutrients, ions, water. There's the passive reabsorption of ions, and there is secretion. There's all of these different events going on, but sodium reabsorption is important in all of them. So this is the key, that sodium is the key to all of the functions that are happening at the proximal convoluted tubule. Okay, you guys following that one? So I'm not going to hold you to all the very nitty-gritty details of that, but the idea that we're absorbing nutrients, ions, water, both passively and actively. And in all those situations, it requires sodium. All right, on to the loop of Henle and countercurrent multiplication. The body is incredible. It is so incredible. I feel honored to be able to talk about this. Of all the things people get to talk about on campus, the human body, it's so intimate. It's this thing that we walk around with every day and so overwhelmingly sublime. It really is. So I feel very privileged to talk about it. And this is just one of them. I mean, we're talking about urine here, pee. But it's really cool how this works. So here it is. The loop of Henle and countercurrent multiplication. So what happens here is perhaps rather, so the flow of fluid is coming down this way. And rather than talk about the first part of it, let me go to the second part first. In this thick ascending limb, this membrane is thick because it is impermeable to water. Water cannot pass. It cannot pass through that. Instead, there are these transporters, active transporters, meaning they're ATPases. They use ATP to pump sodium chloride out into the extracellular fluid, into the interstitium. So we're burning ATP on this side and pumping sodium chloride out. And what happens is it creates, so you see these white boxes down the middle, it creates a concentration gradient. It creates a concentration gradient down along the length of the loop of Henle. Coming back to the other side, the thin, descending limb. So this part of the membrane is fully permeable to water, but it's impermeable to various solutes. So water leaves the tubular fluid, leaves the descending limb passively, going down its concentration gradient. All right, it's getting sucked out of the tubular fluid because of the osmotic gradient that's being created by the ascending limb actively, countercurrent multiplication. So the ATP that we're burning on the right-hand side, it's not only, that energy is not only going to export sodium chloride, but it's also, that energy is also being captured to drive water resorption. So again, sodium is the driving factor here for the physiology that we're observing. A huge amount of fluid is resorbed here. So the way I describe it here, maybe if different ways help you understand it, different ways of saying it. Solute pumping at the ascending limb increases the solute concentration in the descending limb. And is going to accelerate the solute pumping in the ascending limb. So they're self-reinforcing, the two gradients, the two water and sodium reinforce each other's resorption. Because of this is extremely effective. Only about 15 to 20% of the initial filtrate volume is going to reach the distal convoluted tubule. That means most of the water, 80 to 85% of the water that's in the urine is going to get picked up, primarily in the loop of Henle, but some of it in the PCT. Okay. Questions before I move on? Heady day today. All right. So here's some definitions. Let's take a little bit of a break from the pathways for a moment, and let me just give you some definitions, some concepts. There's this idea of renal threshold and transport maximum. So transport maximum is a rate. It's a rate. So amount of solute per time, for example. And it's a maximal rate, in fact, of either secretion or absorption of something, some kind of solute by the renal tubules. Right? So at a certain point, a part of the renal tubule, it just can't move any more solute. The rate at which it's moving solute, that's the transport maximum. Renal threshold is the concentration of solute at which you're going to hit the transport max. It's the concentration of the thing that you're moving that you get to the transport maximum rate, the maximal rate of transport. Does that make sense? So the definition that I have written on the board there is the plasma. It's the plasma concentration at which a specific compound or ion begins to appear in the urine. So if you go above the renal threshold, if you go above, you can't go above the transport max. If you reach transport max and you go above your renal threshold, whatever that thing is that you're transporting is going to end up in the urine. All right? If you're at transport max and the concentration continues to go up in the blood of whatever this thing is, then that stuff is going to show up in your urine. And this renal threshold is different for every single solute. Okay, it varies by solute because every single solute is getting handled by different transporters. All right, so let's look at glucose first. Here's a little should I walk you through the diagram? Should I walk you through that diagram? It's just a pictorial description. So here we are below the transport maximum, and this protein, this black wheel, is the transporter. It's pulling stuff out of tubular fluid back into the blood. Okay, it could be the other way around, but and here it is on the bottom. This is the other way around. It's pulling something actively out of the blood and chucking it into the urine. So at the transport maximum, so you see here below it, there are empty sites, right? It's like not, it's not fully saturated. It's not at its capacity. Am I blocking you? And then here at the transport maximum, every single opportunity it has to make that transfer from serum to urine or tubular fluid back to serum is being occupied. Above transport maximum here, now we're trying to, this is a resorptive process. We can't resorb everything. Some's getting past it from the blood and occurring in the urine. And likewise, here we're able to pull all the stuff out of the urine, but now we're past the renal threshold, and stuff is staying in the blood. So with glucose, it's pretty high, 180 milligrams per deciliter. That's 180 milligrams per deciliter. If you go above that, this is called glycosuria and is indicative of diabetes mellitus. So now you can see why, what was his name? Hippocrates named it diabetes mellitus because he'd take a swig of the urine and it tasted sweet because the person probably had diabetes. They had diabetes mellitus, and sugar was appearing in their urine. We were past the renal threshold. For amino acids, it's significantly lower. It's around 65 milligrams per deciliter, depending upon what amino acid we're talking about. Some amino acids have higher or lower renal thresholds. So because of this, amino acids commonly appear in the bloodstream after a protein-rich meal. Say you are a bodybuilder or an athlete of some sort and have a high protein diet. The truth is you're probably secreting a significant amount of that into your urine. And we call this amino aciduria. The last concept is simply natriuresis. This is the release of sodium into the urine. A lot has been made about the resorption of sodium, but if you have an extremely high level of sodium in the blood, then there can be natriuresis, or the release of sodium. Okay, there you go. Jam-packed day. I tried to not rush through it. I tried to go slowly for you. How was that?